All right, good morning, everyone, and welcome back to Semel Grand Rounds. Today we have Dr. Matthew State presenting on the genetic basis of neurodevelopmental disorders such as autism. Dr. Marta will be on in just a few seconds to introduce Dr. State, but first, a few quick reminders. If you have any questions, feel free to write them in the Q&A at any point, either during the talk or once the questions have begun. Also, please remember that you have to fill out the survey in order to claim CME credit. The survey will either open immediately upon closing Zoom or you'll receive an email tomorrow. And with that, I will turn it over to Dr. Martyr.
Hi, it's a personal pleasure to introduce Matt State. I first met Matt when he was a UCLA resident on my ward at the VA. Later on, he spent a year in the schizophrenia clinic, or the psychosis clinic at that time. And it was very clear to me that Matt was an extraordinary clinician. He was self-assured, compassionate. Our psychosis clinic is always filled with stresses and unimaginable dealings with psychotic patients and family members. And I, at that time, predicted that Matt's self-assurance and his ability to bring both knowledge and compassion to treatment, that he would emerge as a leader in psychiatry. Following his residency, he completed a child fellowship at UCLA, and then he did something that none of us would have predicted. But after finishing his residency, he decided that to understand the developmental disorders that he was seeing, that he really needed to learn genetics. So he decided, with very little experience in research, that he would get his PhD in genetics. He went off to Yale, where he eventually got his PhD, joined the faculty, and began an extraordinary career as a genetics researcher. He's headed a number of large genomics coalitions that focused on childhood disorders such as autism spectrum disorder and Tourette's. In 2013, my initial prediction that he would become a leader in psychiatry became true, because he came back to California and became the chair at UCSF. He's currently the Obendorf Family Distinguished Professor, as well as the chair and the president of Langley Porter. He's won numerous awards. In 2017, he was awarded the Cernat International Prize in Mental Health. I believe he's going to talk to us about how the detection of rare mutations of copy number variations and large de novo mutations can actually be tools for learning about the biology of illnesses such as ASD. Again, I'm very pleased and proud to introduce Matt State. Thank you.
Thank you, Steve. It's such a pleasure. I'm thrilled to be back, even virtually. I had hoped that this would be in person and have an opportunity to come and say hello to old friends. UCLA played an enormous role, obviously, in my professional life beyond training. I was just mentioning to Steve, I didn't do a psychiatry internship, so my very first experience with psychiatric patients was on Steve's ward, and enormously impactful. I would not have been a child psychiatrist if not for my rotation with Mark D'Antonio. Completely changed my life and turned me into a child psychiatrist. And I wouldn't have gone to Yale for a PhD if not for Dan Geschwin putting me in touch with the folks at Yale and really encouraging what at the time seemed like a pretty out there idea after completing clinical training to start from scratch with learning genetics. And all of that has landed me in a place where it's been tremendously rewarding. And I want to share with you kind of a perspective of the last 10 years or so of progress in gene discovery and autism. When we started this effort in the lab, it was extremely little, almost nothing was known around the genetic contributors to common forms of ASD. And now there's been substantial progress. So what I want to do today is talk relatively briefly to summarize the success in gene discovery in autism spectrum disorders, give you an idea of where we've come over the last decade, and then spend a good deal of the talk, as Steve just mentioned, really talking about the next great challenge for us now that the field has been quite successful, as I hope I'll convince you momentarily, that the challenge now is how do we take that and make it have an impact on the lives of kids and families by understanding pathophysiology in a way that helps us develop novel somatic treatments that do a better job of attacking the core symptomatology of autism, the social and impairments, and other associated phenomena. And then I'll talk briefly at the end about sort of where we've landed now. The middle point that I'm going to make is that this idea of going from genes to treatments, that there's an important distinction here that genes obviously drive biology, but that's not precisely what we're interested in. You can learn a lot about the biology of a gene and still not understand how it's contributing to mechanisms of disease. And so there is this fundamental challenge, I think, again, I'm going to spend some time on this, of distinguishing the biology, which we can begin to see almost immediately as soon as we identify a gene, from the pathophysiology, the thing that we hope will drive the development of novel therapeutics. Before I move on, I do want to say, that throughout this, I'm going to talk about work in the lab and some of the large consortia that Steve mentioned. But this is so bringing coals to Newcastle to come back to UCLA, not only, for the reasons that I described previously about all of the impact that folks have had on my career, but also in the field of autism. It really is not hyperbole to say that the Geschwin lab at UCLA and generations of trainees probably, I think, have had more to do with shaping the field of autism research, biological autism research, than any other place really in the world, from the very earliest stages in pushing for collaborative genomic studies and making genomic materials and data available to the entire community, which may have been the single most important transition to moving from really abject failure in gene discovery to a decade of success, all the way through introducing transcriptomics to thinking about how to understand the differences in autism brains. So as I talk throughout, there are going to be strains of all of that you'll see influencing. So even though it may be talking about my lab or the Autism Sequencing Consortium or whatnot, it really, UCLA and Dan and his trainees have played a huge role in what I'm going to talk to you about. Because I have a tendency to run on, it's gotten worse as I've become a chair. I want to start by identifying the people who've really done the heavy lifting here. As always, PI is only as good as the folks who are at the bench in the lab full time. And two folks in particular, Stefan Sanders and Jeremy Wilsey, are graduates of my lab, both as doctoral students and then as postdocs. They are now running their own laboratories here at UCSF, doing remarkable work, really taking the lead in major areas now of autism, genomics, and biology. You'll see their names throughout, but none of what I'm showing you would be possible without their intellectual contributions. We have, it's like woebegone in my lab, thank goodness. All of my postdocs are, just stellar. I'm going to mention a new paper from Helen Wilsey, who has come to the lab and started a Xenopus laboratory to help think about how to make progress on differentiating biology from pathophysiology. All right, so as I said, I'm going to summarize the where we are in the genetics of autism, and really tell you, as Steve mentioned briefly, sort of three large categories of changes in the genetic code that represent sort of the largest group of now well-defined, say, definitive genetic contributors to autism spectrum disorders. So one, Steve mentions copy number variations. These are submicroscopic changes in chromosomal structure. What we've identified in autism is that the loss or gain of copies of one of the two pairs of chromosomes that are inherited is sufficient to lead to dramatically increased risk for autism and a number of other neurodevelopmental disorders, a topic I'll mention briefly, but just to have that in your mind, that we're not talking about the total absence of a section of DNA, but the loss of one of two or loss or gain at one or two chromosomal pairs. And the kinds of changes that we're going to be talking about are typically very rare in the genome, and many of them are not inherited. So these are new, they arise de novo in the germline in sperm or egg, typically just prior to fertilization, and lead to the presence of a mutation in the offspring when that mutation could not be found in either of the parents because it was spontaneous in sperm or egg. A second category that's been tremendously important and informative for the field is the identification also that these rare de novo mutations can involve just a single change in the letter code of DNA at a single base or nucleotide. We call these single nucleotide variants. And the focus of a lot of that progress in gene discovery in autism is around looking for these kinds of rare de novo mutations in the 1.5% of the genome that actually codes for genes. Those are kind of the business end. As you'll recall, genes coding for RNA and then often coding for proteins. And so what we're looking at here are things that are actually hitting those units of biology. There's a third area where there's been tremendous progress across complex disorders in psychiatry. In autism, it hasn't been quite as robust in terms of the number of spots in the genome that have yet been identified for reasons that I'll describe for you. But those are, I think, pretty familiar to everyone in the audience. They're called single nucleotide polymorphisms, or SNPs. These are also typically changes at one a spot in the genome, sometimes maybe a little bit bigger than that. They are common in the population, meaning that they are present in more than 1% of the general population. Unlike the other kinds of mutations I'm describing, they're transmitted from generation to generation. And often these are found outside of genes in what we call the non-coding region of the genome, and to a great degree in portions of that non-coding region that help coordinate or regulate the activity of the genome. So just to give you kind of a visual on how this plays out and the story in autism, as I've told you, many of the mutations that we've identified, both in the structure and sequence of DNA, involve these new or de novo mutations. The diagram that I'm showing, I'm wondering if I can get a pointer here. How do I do it? Let's see. Annotate, maybe. Mouse, that's good. And I'm going to go back one slide, I hope. I guess I'm having trouble going back. Let's try here. Okay, technical problem solved. So showing here a diagram of a family structure in which neither of the parents are affected. There's a single affected offspring, and if there are siblings, they are unaffected as well. The idea here was that if we found families that look like they've been hit kind of by lightning with regard to the presence of a disorder, in this case autism, it may be because there was a spontaneous mutation in sperm or egg affecting the germline and causing a de novo mutation that appears again in the affected individual but is not present in unaffected members of the family. There are reasons that those kinds of mutations, just theoretically, if you were trying to find a change in the genetic code that had a large impact on a disorder, an early neurodevelopmental disorder, these mutations can have that relationship, in part because there's very little time for natural selection to act on the mutation once it emerges. As long as the fetus comes to term, you can imagine that you could have very significant impacts on things like social functioning, intellectual functioning, even brain structure, that could in the long run make it difficult for that genetic material to be passed generation to generation. But in the first generation in which it arose, you potentially could see a relationship between a mutation and a very large effect, where if you waited 4 or 5 generations to try to see that mutation, likely it would have been purified out of the genome through natural selection. So as I said, this is just from the NHGRI, and it's chromosomal abnormalities. And as I described for you, copy number variations are really just chromosomal abnormalities that fall below the resolution of a microscope. And so we use other methods to detect them, but they basically are the same idea, that there changes either the loss of a segment of DNA, we call it deletion. There can be additions of segments of DNA on one of the 2 chromosomes in the pair. This can be a duplication, or if there can be more than 2 copies, and we call it an amplification. And one of the most important kind of seminal early findings in autism spectrum disorder, several labs, but really focusing on autism in these kinds of families, were Mike Wigler and Jonathan Sabot at Cold Spring Harbor. And they focused on whether or not there was an increased rate of these submicroscopic chromosomal changes de novo in affected versus control families. And they found that there was in a quite small sample of individuals for a genetic study, less than 200 people. By 2015, Stephen Sanders, who was at the time in the lab, led the publication of a manuscript that reached more than 2,000 families, and validated this finding that there's an excess rate of these de novo copy number variations, both duplications and deletions. In our study design, we were able to compare the affected individual to their unaffected sibling, which is just a beautiful, elegant genetic experiment to be able to carry out, thanks to the Simons Foundation that collected, as I said, several thousand of these families. So the lion's share of gene discovery with regard to autism really built on that finding, and it was enabled by the advance of technology that allowed us to go from segments of the genome that may be 50,000 or 100,000 base pairs down to the individual letter code, to individual bases of DNA. This is from Stefan, once he was out of the lab, wrote a really lovely review on next generation sequencing in autism, again, demonstrating that we were using these families with ASD cases versus controls, looking for de novo mutations, extract DNA, capture the portion of DNA that corresponds to the coding portion of the genome, as I said, about 1 or 1.5% of the total human genomic complement. These are then sequenced through next generation sequencing technology developed just about the time we were able to take a first look at these autism families. And then there was just a comparison of the rates and cases versus controls in order to identify whether or not there's risk associated with these mutations. And this is, again, when Stefan was in the lab, multiple laboratories landed on this answer simultaneously. So Mark Daly's lab in Boston, Evan Eichler's in Washington, Mike Wigler's in Cold Spring Harbor, and ours published within, well, 3 of us in the same issue of the same journal, one several weeks later, showing the same thing, which is that these mutations are overrepresented in cases versus controls. And that, let's see if this works. Yeah, great. So that the strongest signal in terms of effect size were for mutations that were damaging, predicted to be damaging to the encoded protein, meaning that the change in the letter code led to something that could be inferred to either completely eliminate the product from that copy of the chromosome, or to damage it so severely that it would be gene disrupting. So we call these likely gene disrupting mutations. It turns out that we were able to see initially a somewhat less robust signal in terms of effect size, but these are what's called missense mutations. So they're mutations that don't completely kill or damage the protein, but can lead to a change in function. So what we're seeing is that the mutations are are not uniformly problematic. These are mutations that would not be predicted to change the function of the gene, and there's no discernible difference between cases and controls. It is these likely gene damaging, and the more damaging they are, the more effect that they had, which really drove the notion that these individual changes were carrying autism risk. Now, how do we go from that to identifying specific genes? Basically, we just look at where they add up in the genome. Again, this is from Steffen's Cold Spring Harbor review, which is a very nice diagram, I think, that just shows the actual data currently around where mutations, these de novo mutations, are lining up in genes where we see a dramatic overrepresentation in cases versus controls. And there's some really elegant statistical advantages that come from looking at de novo mutation I won't have time to talk about now, but perhaps in the Q&A if people are very interested. This gives us tremendous statistical power to say that SCN2A specifically is a gene that's carrying risk through a consequence of the de novo likely gene disrupting mutations in the coding portion of the genome. And this takes us to kind of where we are. So I've summarized now really a decade of work looking at rare and de novo mutations, which has really been the greatest area of progress in autism genetics. And this is a summary from 2015, but still a pretty good snapshot of the copy number variations that have very strong statistical evidence for association with autism. And then on the right is a recent paper in 2020 from the Autism Sequencing Consortium involving our lab and Joe Buxbaum's lab and countless others across the country, including at UCLA. And collectively, this is now at 102 genes that have statistical evidence for association with ASD, and about 36 that exceed a family-wise error rate correction, which essentially is genome-wide significance. So, so the world is now filled with not only regions of the genome, but specific genes that have extremely strong statistical evidence for association with ASD. Before I move on to talk about biology, I just want to point out that the work looking for kind of the other end of the allelic spectrum, if you will, looking for the contribution of common transmitted polymorphisms in the genome through what's called genome-wide association. Again, extremely successful in bipolar disorder, schizophrenia. Now they have 250 alleles that have been identified in schizophrenia. The story in autism has been somewhat different. And here, with a sample size now of 18,000 cases and 27,000 controls, there are 5 regions of the genome that meet a statistical significance threshold that's widely accepted in the field. And you can see the list here. Again, this is for another talk, a really interesting question, not only about sort of the overall genetic architecture of autism, why it might look like this, why it might look different than schizophrenia or bipolar disorder in terms of the distribution of these rare versus common alleles, but also really interesting, I think, potential opportunities around combining what we're now learning about common transmitted variation in the genome and how that might interact with the kinds of rare mutations that I've just described for you, which, while they represent the majority of what we've discovered in terms of individual genes, represent somewhere in the neighborhood of 20% or so of the clinical population that comes in, to an autism clinic now. Ultimately, I think when all said is done, it will be somewhere in that neighborhood for the combination of copy number variations and rare point mutations. This slide is the last one I'll do on genomics, and this is actually from a study of schizophrenia that has just been posted to the Med Archives, and will, I'm sure, be out soon. It's from Mark Daly's group in Boston, a large collection of other laboratories. And this shows a bunch of really interesting things. So what this is showing is the effect size. So how big an impact different kinds of variations have. Here are copy number variations. Here are 10 individual genes that have been discovered in essentially the identical way that we've identified 102 autism genes. In schizophrenia, again, it's been the kind of the mirror image story where they needed very large samples to get to the first 10 of these rare mutations. And these are the genome-wide hits in GWAS. in schizophrenia. And so what this is showing is that the common alleles have very, very small individual effects, that some of the CNVs and essentially, and many of the point mutations have very large effects. So we're talking 20 to 100-fold increases in risk here. And the autism genes, the mean effect size for the rare mutations that I talked about in the most statistically significant group are above 20-fold, or have a mean of about 20-fold increase in risk. So these are very large. But there's another interesting thing here, which is that when you look at this list of CNVs, these are all schizophrenia. These are essentially they are the identical list. When you look at, 2 slides back, Stefan, the paper that Stefan was lead author on, demonstrating where we were in autism in 2015, and still, pretty much that's the group. Tremendous overlap, no specificity here. And in fact, these not only increase risk for schizophrenia and autism, but also intellectual disability. Some of them increase body weight, epilepsy, tremendous overlap, which creates some significant conceptual challenges for thinking about how you dig into the biology to understand how to treat ASD if you're relying on CNVs because of the wide variety of phenotypes that may emerge. Now, consequently, and this is just emerging data, of these first 10 genes for schizophrenia, while there's tremendous overlap here, there's only a single gene out of these 10 that overlap with the highest confidence autism genes. And if you move down to the next 20 or so genes that didn't quite make it into this group in schizophrenia, there are only 3 total that overlap with autism. So that begins to suggest that while there may be, a tremendous difficulty with specificity here, while there clearly are broad phenotypic implications of the rare mutations that I've talked about, where there's obvious overlap with intellectual disability, in some cases epilepsy, I think this is beginning to suggest that there might also be some degree of specificity, at least with regard to early versus somewhat later onset neurodevelopmental disorders, which I think will be quite interesting in addressing the next set of challenges. So I think I've been encouraged to have a half an hour, so I think I have about 10 more minutes. If anyone wants to raise a flag and tell me I have less than that, please just jump on and let me know. So I want to talk to you about the challenge of how you get from this kind of static list of genes, it looks impressive, 102 genes, to some understanding of biology. And you might think, oh my God, that we found already 102 genes. Actually, we can predict that just based on de novo mutations, that there are going to be 500 to 1,000 genes that may be contributing through that mechanism. Does that mean that we're going to have to be stuck trying to dissect 1,000 different biologies or 1,000 different pathophysiologies in order to be able to come up with novel therapeutic approaches? And I think from the very earliest successes in gene discovery, at least we had hints that may not be true. And what's happened, the reason that we had that is really from the start and now through the entire wave of gene discovery over the last decade, is that there have been 2 really large areas of biology, despite the great diversity in the specific molecules that have been identified and their presence in every compartment of the cell. There have been at least 2 large groups of function. So one having to do with the synapse and both presynaptic and postsynaptic molecules, a lot of postsynaptic molecules. And then a second is around the regulation, coordination of gene activity or gene transcription. And here in particular, a whole group of what we call chromatin modifiers that alter the conformation of DNA to dictate whether or not it facilitates activity or depresses activity, et cetera. So there was insight right away, I think, and with every additional wave of gene discovery, this has been reaffirmed, that these 2 large categories fall out. They're not exclusive. There are other things, and I'll talk about them in a minute. Now, this, in some respects, is great news. I have to say, when I started in the genetics period of child neurodevelopmental disorders, but autism in particular, I had, diagram envy, because if you went and you took a look at the genetics of heart disease or immunological disorders or whatever, they always had these beautiful diagrams that showed how the molecules interact. In autism, we had no genes. So simply now being able to show you a diagram that shows relationships between the protein products of these genes and that they fall out into these different categories is exciting. On the other hand, if you're a skeptic, you could take a step back and say, wow, you spent 10 years, an enormous amount of time trying to understand a neurodevelopmental disorder, and what you've identified as a large group of genes involved kind of in neural and a large group of genes involved in development. And I think that that's a little bit of a facile critique, but it's not completely misplaced, because the reality is that while these fall into these groups, they really kind of at first blush point to just such a wide swath of biology that the notion that we're going to be able to get from here to being able to develop novel treatments remains a significant challenge. And I just want to take you conceptually through sort of how it is that that sort of our early ideas about, well, once we find a gene, it's going to give us a path to identify biology and treatment options is complicated by the what we call the pleiotropy of the individual molecules, that they don't necessarily do one thing, but multiple other dimensions of trying to get our hands around the biology of dozens to hundreds of genes contributing to a single disorder. So I would say that, naively, as I first set out to try to find rare mutations in autism, the model was this, that we would find a very rare mutation, that we would be able then to know what the protein was, to map it to cell type, understand then what it was doing functionally, and then we would use that to develop a treatment. And a lot of this was driven by my neighbor and mentor, Rick Lifton, who had done this with regard to blood pressure regulation and kidney function. And, in retrospect, once I discovered genes, it was embarrassing to some degree that as a child psychiatrist, that, one thing that had completely escaped me in building this model of success was the complexity of the development of human brain, which you would think would be at the top of my list of things to think about. But let me take you through kind of how that plays out here. So as I said, the biology of the individual genes that we've identified are generally pleiotropic to some degree, in the sense that it's very common for a single gene to lead to multiple different protein forms, and those forms may, particularly in the brain, operate in different brain cells and even types of brain cells, comprising multiple different circuits in the brain, and then leading to a variety of behaviors. So even if you know a gene, trying to figure out which pathway, whether it's all of these pathways or one pathway that gets you from genes to some critical component of pathology, is uncertain. But, again, what we're talking about is the human brain. So not only is there complexity around cell types, et cetera, but this is a developing organ of enormous complexity. And so when you start adding just the notion of space and time, that these genes, as they're unfolding in their activity, are doing that along, in different ways, regionally, in terms of, when a particular version of the protein at what time it is being expressed may impact what cell it's found in, and that may impact what circuit it's contributing to, and whether it's the development of that circuit or the function of that circuit, then leading to complex behaviors. So just adding space and time gets you into this enormous search space from genes moving on. And then we have this problem that if we study that in mouse, it's very likely that the things that are most conserved to human are going to be closer to the gene, and that the further that we get towards the complexity of human brain, it's not that it completely falls apart, it's just that it gets harder to feel confident. You can measure pretty easily how similar a mouse gene is to a human gene, but getting here raises a number of additional questions. So that then gives you a sense, and then actually I have to add the complexity in autism. So you have the developmental trajectory of human brain. So you have space and time, you've got the pleiotropy of individual genes. And then in autism, you have sexual dimorphism. So I can't draw another dimension, but add that here to trying to figure out how you get from an individual gene and wind your way through a pathway where you don't get lost in an impenetrably complex biology and trying to figure out what you need to target in order to change a behavior, and that behavior change or electrophysiological change or some other change in mouse then also needs to map onto the things that we care about in human. So one of the ways that we thought about dealing with this actually was hinted at in that previous diagram that everyone did, I think, pretty intuitively at the start of the process, but we added to it and actually really in absolute stride for stride with the Geschwin lab at UCLA was to say, you know, maybe the way to begin to think about how you could differentiate the pleiotropic biology of these individual genes to something that is more critical to the unfolding of autism in the developing human is to say, let's take advantage of the fact that we have multiple swings at this. It's not just a single, you know, the Huntington's gene, and we've got to find our way through this. We've got now initially, you know, a dozen or so, now 100. And then you could ask a slightly different question, which is in all of this complex biology, if you're able to trace it, not just in terms of what genes are doing, but when and where they're doing them, that you might be able to narrow in on some of the critical parameters to study pathology. So if you could go from genes to understanding what cell type brings together multiple autism risk genes, potentially not all of them, but say a significant subset of them, that could help you then design experiments that could begin to, you know, serve as guideposts as you try to move from genes to biology. So here, just keep having trouble with, we initially tried this in 2013 in collaboration with Ninad Sestin, who was developing maps of the trajectory of gene activity across brain development. If you think about what I just described, what we needed to do was to take the individual genes that we had identified and somehow use them in order to trace the activity of genes as they unfolded in developing the human brain. And so Ninad helped create this map. It's now called the BrainSpan Project. This is multiple brain regions that were dissected post-mortem from 4 weeks post-conception all the way to 80 years of age. And then we used one approach. There are multiple approaches to taking our individual genes, looking at their expression, asking questions about what other genes are they expressed with the hypothesis that if genes are expressed together, they may well be doing something together functionally. That's something that I think has proven out to be the case in multiple different kinds of experiments. And so we took every high confidence autism gene, looked at their expression patterns, created this matrix of brain regions and time to try to kind of capture this picture of the unfolding of the human brain around these autism risk genes. And then we asked the question, if we took a second set of genes that we knew were related to autism risk or had a high probability of being related to autism risk, did that pick out any place and time in the developing human brain that appear to be overrepresented for autism risk in a statistically significant way? And we were able to show in 2013, again, we published in the same issue with Dan Geschwin, who took a conceptually similar approach, but using somewhat different gene lists, different analytic approaches to thinking about gene expression. And we both landed on the finding that there was a statistically dramatic signal in mid-fetal development of the human cortex. We got different answers as we pursued that down the line, or slightly different in terms of exactly what kinds of cells and in which layers. So we found that these were glutamatergic excitatory neurons in the deep layers of the human cortex. Dan found the greatest degree of convergence around the same types of cells, glutamatergic excitatory neurons, but he found them in upper layers. We now have layer 4, I think in particular, and we've been able now to repeat this with increasing number of genes. This is unpublished data, basically showing, showing the same thing. I think in part also showing that there are likely to be other regions. This is medial dorsal nucleus of the thalamus and the cerebellum, and there was a bit of a signal there, which I think still needs to be pursued. These are limited to some degree by the underlying expression data, which is an important limitation. You can only kind of see what you're looking at. So if you're primarily looking at cortex, which is what primarily we were able to look at, the fact that there's convergence there remains an important observation, but it's not exclusive of other points of convergence, which we would expect in a disorder as heterogeneous as autism. And this is just a conceptual picture showing what we did, taking the mutations with brain expression data, identifying an expression network, mapping that to the development of the human brain, and then in our case, finding that these deep layers of cortex were a point of convergence. We actually now are just publishing in Neuron, it's in Press. This is a paper that is taking on different issues, but looking for convergence using a model organism, Xenopus tropicalis. But as part of doing that, we went back and did another similar analysis now of the 102 high confidence autism risk genes, again, looking at their expression, how they're expressed together, and then seeing whether we could, map that onto layer specific information in developing human brain. In this case, we focused on mid-fetal cortical development because of our prior findings to see if we could get some greater resolution about where we see this convergence. And in our case, we recapitulated our finding that in the inner cortical plate, so what will become the deep layer cortical plate or deep layer cortex, but we also found evidence of convergence in subventricular zone and subplate. And the subventricular zone in particular is consistent with some of the other findings in this new paper around another point of convergence in addition to synaptic function and transcriptional regulation, which is neurogenesis. And again, this is something that's been kind of percolating up through the literature in a variety of different ways, including a lovely paper a couple years ago. I keep pointing to Dan, and it's only because always seems to be right where the action is, looking at the early development of the cortex and the contribution of genetics and genetic risk to neuropsychiatric disorders around this process of neurogenesis. So I'm going to finish here, I think, by giving you, because I think I only have 3 minutes, by just kind of summarizing the good news, and then I'll take a minute to talk about it's not all, I think it's generally good news, but in any story, there's clearly lots to be done. And I should have started by saying that, while I think it's been a remarkable era of progress, we still have not fundamentally changed how we're dealing with kids in clinic from the time that I first got interested in doing the genetics of neurodevelopmental disorders. It's not to say that we're not helping kids in many ways, but from the point of view of of using biology to transform how we're treating, we're moving too slowly for families and patients and clinicians. But just to kind of fill in where we are, again, this is kind of a similar diagram of this kind of complex biology going from genes all the way to behavior and the difficulty in sorting through that, and then potentially the use of convergence. So we now have both rare de novo large effect structural and sequence variations that have very large biological effects, and we have common alleles that will underlie the vast majority of population risk, each one though carrying very small increments of risk. There are multiple pathways that have been identified from this, looking at individual proteins and how they work together. A number of the canonical developmental pathways, cancer pathways. A very striking finding in every study so far is that the autism genes converge around being targets of the Fragile X mental retardation protein, which just opens up a whole kind of area, I think, a very interesting discussion about the overlap of what we, sort of separate out as these monogenic neurodevelopmental intellectual disability syndromes and what we're seeing in clinic with common forms of ASD. This now converges around chromatin, transcriptional regulation, synaptic function. This is not an exclusive list, but from a standpoint of what the genes are doing, these come up repeatedly. And now neurogenesis, I think, is bubbled up. And as I said, we have a new paper. It's it is online. I have a link I can share with all of you if you're interested, in the journal Neuron, looking for convergence and finding evidence in neurogenesis, both in human and in Xenopus tropicalis. These have led to some ability to begin to define where this risk is converging. Exactly what that means is still an open question, but mid-fetal cortical glutamatergic neurons, both deep and upper layer, have evidence for contribution. Now, additional evidence, I think, around neurogenesis, particularly in early mid-fetal development. And there's emerging evidence as we get more information about the developing human brain and kind of foundational resources around the importance of the striatum, leading to what may seem like not a huge breakthrough, maybe a penetrating glimpse into the obvious that these are cortical striatal circuits. But again, I think you can appreciate that this is a tremendously filled out kind of notion of where we are in the field, where when this started 15 years ago in our lab and the Geschwin lab and multiple others, these boxes were pretty much empty. That is going to have significant impacts on our thinking about treatments. And because of the time limitation, I think I'm only going to focus really on this last point, which is that this does give us an opportunity now with new tools to begin to think about something that, maybe would have been considered entirely implausible. This is already underway now in syndromic, quote unquote, monogenic syndromic disorders like Fragile X and Rett syndrome, et cetera, to think about targeting nucleic acids directly. This is, I think, enabling traditional pharmacological approaches, using both individual genes and trying to rescue their impact, as well as using these convergent approaches. And again, I want to highlight Helen's paper, which I think is a really, really lovely example of using multiple model systems to identify not just risk, but potential resilience factors. But the last point I want to identify is that, what you've seen is the product of, of an enormous investment by NIH, the Simons Foundation, and parent advocacy groups to drive forward human genetics. And I would say that, in my view, the key now to making all of this possible is the same kind of massive push on understanding the clinical manifestations of early development in a way that we simply don't have our arms around now. That measuring change, particularly understanding early developmental trajectories, what are reliable markers of deviance from a normal developmental trajectory, are going to be absolutely critical for success in leveraging any of this biology, just kind of pointing to the absolute necessity of bringing together clinical research and basic research in order to make progress in these areas. This is the lab. I've highlighted the key folks at the start of the talk again, because I always know that I have a tendency to run late. And I want to thank you for your attention and hope I've left a fair bit of time for questions. Thanks.
Thank you so much, Dr. State. That was a really excellent talk. And we have had a few questions that have come in. I think I'll start with the question. In the talk, you largely kind of focused on the idea of genetics as contributing factors towards autism. And I think especially in the media these days, there's a lot of thoughts about like environmental inputs, with perhaps like vaccines being one of the ones that gets the most media attention. And I'd just be curious to kind of hear your thoughts on the differential contributions between genetics and potentially even environmental inputs as well.
Yeah, absolutely. It's a great question and a really timely one. So there's several parts of this. So one is the nature nurture debate or dichotomy, which isn't, I have to say, even describing it as a debate, and almost everything you see in the emergency room, there's some combination of nature and nurture, right? And neurodevelopmental disorders, psychiatric disorders are no different. So for autism in particular, I think, we have some traction on the notion about sort of the relative contribution. It's not all genes and it's not all environment. If you parse it out and you look at twin studies and other ways of trying to divine, how much genetics versus how much environment, genetics has a tendency to be overrepresented, environment underrepresented. And in fact, when you look across medical disorders, autism is frequently, almost universally at the top of the list in terms of genetic contribution. So certainly among psychiatric disorders, but really among all common disorders that come to the emergency room, the overall genetic contribution is quite large. And people put a number on it, and there's a debate: is it 60%, 70%, 80%? The studies in general, I think, have a tendency to coalesce around the higher number, but there have been some very good studies that have called that into question. In my view, there are a couple. So that's one thing which I think is important to point out. The second is that with regard to vaccines, there's a ton that we don't know. In fact, there's very little that we know about what significant environmental inputs are to autism risk. In some ways, it's because while the genetics is difficult, the genetic code is finite. It's got 4, component bases. As you can see, we can narrow in and do a really reductionist kind of experiment and get down to very specific factors, something that's been hard to do in large scale epidemiological studies. But one thing that seems really clear is that vaccines writ large are playing no significant role in autism risk. And it's because there's been an enormous amount of energy and literally millions of kids in studies looking at vaccines and looking for some signal. And there really is no reliable signal there. So I think while I can't tell you a lot about what it is, I feel confident in saying what it isn't. As a clinician, what I would tell my patients always is that there's nothing more important to me than my daughter Hannah, and I didn't have a second's hesitation about her, after spending years looking at the biology of autism, about her being vaccinated. The final point that I make really has to do with what I just said in terms of what I think is solvable and in what order. Part of the reason that I got really interested in genes was precisely because, and rare mutations, it wasn't because it was going to explain all of autism. In fact, when I started out, like my common talk before we were successful in gene discovery was, if I can find one gene that I feel confident is leading to autism and use that to get an entree into biology, that will be something valuable because of how little we knew about the actual molecular and cellular underpinnings of disorder. The 102 and what we've learned is like beyond sort of anything that I thought we would potentially be even near in terms of contribution to the field. But it is a reductionist approach. And I do think it's an avenue to say, look, if we can understand something quite concrete about a very complex and heterogeneous disorder, that is a way to make progress. And ultimately, I think it is going to be a way to open up a deeper understanding of what environmental inputs are. And it's in fact very likely that significant environmental inputs are going to be easier to manipulate in the long run, in terms of population health, than, other kinds of prevention, to the extent that these are genetically driven. So I think it's all important. And I think the battle around, is it genes or is it environment, is a little bit of a specious argument, but it is really important, I think, to be able to give people confidence that it ain't vaccines.
Thank you. And I think that segues pretty well into the next question that we got. Someone in the audience had asked, first, really great talk, thank you. And then asking, what would be the big picture change in terms of evaluation and treatment of autism from the perspective of genetics? Are we looking at things like polygenic scoring of patients or potentially even parents to assess for risk?
So I think one of the things that I've showed and could have been the entire talk was sort of how much we've learned about the genetic architecture of autism. And right now, in terms of where we are, as I showed you, the contribution that we've been really able to define for common variation falls way behind adult onset disorders. And it's very interesting, I think, to speculate about why that's the case. But so as you see kind of polygenic risk scores, increasingly people thinking about how do you use those to try to stratify risk or treatment response, et cetera. For schizophrenia, much harder at this point in autism because we just don't have the significant portion of risk explained by common variation yet. And that's likely a consequence just of the power of natural selection for disorders like autism and intellectual disability. That doesn't mean that down the line, these things that whoever just asked that question, that those won't be part of the clinical armamentarium. It's just that, but it is a reason, I think, when people say, well, God, you got 100 genes, why do you need to do more? There's a lot to be done, not only around polygenic risk scores kind of writ large for autism. But I do think that there's a very interesting story emerging around the intersection of these high risk variants and this polygenic background in terms of determining why one kid who has a mutation may end up with schizophrenia, another with epilepsy, and a third with autism. And that's going to be, I think, potentially quite important in understanding this. In the short run, the clinical utility of genetics, it has, has started to enter practice. It's not huge, but there's an increasing move to go ahead and identify the 10 or 20% of families who have even these de novo mutations for a variety of reasons. And I think, the American College of Medical Genetics has come out, and I think the Pediatrics Society will do the same thing and say it should be part of the workup of any child who's coming, irrespective of intellectual functioning, for autism, because there's sufficient finding of a specific abnormality that even if it doesn't at this point tell us how to treat, it is potentially going to aggregate in a way that will allow people to get additional information about the specific disorder they have, benefit from ongoing research, participate in ongoing research, and bring a sense of understanding to families with autism. Some kids, as a clinician, I explain to families that this, very unlikely to have any impact on how we're able to treat or what the outcome will be in this generation. There's still tremendous interest, not only in contributing to research by going ahead with genetic testing, but also just to know, right? To have something, as opposed to, we have no idea how this happened or why it happened or what's going on, really. I think that does, for some families, seem to have a pretty significant impact on how they experience what is clearly a marathon for severely affected individuals.
Yeah, absolutely. Another question that had come in, can you comment on the apparent overlap between autism and mood disorders, particularly depression, based on work that's come out of Jordan Smoller's and other people's labs?
Yeah. So again, really great question. And it goes back to, as I said, giving a talk on sort of what we're understanding about genetic architecture is so interesting right now. And so there are a couple things. One is that I showed you that slide, and I'll get to mood disorders in a second, but I'll show you, I showed that slide that showed that, for CNVs, like complete overlap across basically every neurodevelopmental disorder. And then you move to these individual genes, and now for the first time, we know that there's overlap, but I think pretty much anyone would say that it's pretty surprising that out of 30 or so kind of top genes for schizophrenia, there aren't overlapping. And in part, that's because when you go to the far right of the left of that diagram, or whatever, where the common variants are, there's tremendous overlap in at least so far we can divine without knowing the specific alleles, but there's a very significant signal in overlap between autism and schizophrenia with regard to common alleles. And so, what this is doing is we're sort of like, and there have been, I think, surprising overlaps in looking at, at how different psychiatric disorders overlap from a genetic standpoint. I mean, we know clinically that there are increased rates of depression and anxiety in kids with, what used to be called Asperger's syndrome. Looking at family studies, there are increased rates in family members of a wide variety of psychiatric pathology. So I guess to some degree, it's not shocking that when you look at these polygenic disorders and ask, of these really disparate disorders, how much overlap is there? And we see probably more than maybe some would have expected. I actually think that the most interesting part of the story now is how, for different kinds of genetic mutations, it looks like there may be some mix of specificity versus kind of more global risk for a wide variety of psychiatric disorders. And I think both of those are really important observations. And they both, I think, may be helpful in leading to different uses of genetics. So on the one hand, if we really are able to get to molecular specificity in autism, there may be individual treatments about an individual gene or groups of genes, based on it's dramatically increasing risks, say, for autism, but not schizophrenia. And the reason that's important is that, like, when do you intervene, right? So in that case, the idea that that the minute that you started seeing symptoms, if you had a real sense of that specificity, that it might give you confidence that it's worthwhile intervening early there. But I think on the other end, as we disaggregate this, as I said, the ability to use polygenic risk to understand course, treatment, response, natural history, I think are all really exciting. And it sounds like a commercial message, but I think there has been a sense at NIH that, look, we're done with gene discovery. Like, you got 100, you don't need 200. And I think that's, I'm glad that there are people out there like Broad pushing on the common variant side of this, because, and being successful in that, because ultimately I think there's still a tremendous amount to be learned, particularly around how we can apply this to the clinic, that will go from individual therapeutics development to these other kinds of insights that I've described.
Well, thank you so much, Dr. State. We're at the end of the hour, so
Yeah.
I'd wrap things up. But just wanted to say thank you for a great talk and for answering the questions, and really appreciate you being here.
Yeah, it's been really an absolute pleasure, and I wish I could see you all, and I look forward, I hope, to coming back when the Earth starts spinning on its axis again, and we can see each other in person. Thank you for the invite. Take care, everyone.
Thank you. Bye.
Bye.